Our presentation is based on a research paper called Specific Silencing of the REST Target Genes in Insulin-Secreting Cells uncovers their participation on beta cell survival. Beta cells in the pancreas are responsible for secreting insulin, the hormone that regulates blood glucose level. Type 1 and type 2 diabetes are caused due to deficiency of this hormone. The absence of repressor element 1, silencing transcription factor, in beta cells is responsible for initiating the gene expression. A repressor is a DNA-binding protein which regulates the expression of genes. It binds to the operator, blocking the RNA polymerase and prevent transcription of the gene. The REST target genes have been widely researched for their functions of neurotransmission in neural context. In order to explore their role in pancreatic beta cells, a transgenic mouse was generated as shown in the first figure. The gene expressed transcriptional receptor REST specifically in beta cells, which allowed them to identify a wide group of uncharacterized genes that contains REST, which binds complementarily to repressor element RE1. Earlier, it was discovered that REST targets are essential in insulin exocytosis. In this experiment, a high level of REST mice were generated. This silenced their transcription of anti-apoptosis gene. A low level of insulin was produced, which indicated high levels of cell death. It was known that under controlled conditions, cell death was also induced by cytokines. This was due to the adenoviral transduction in insulin cell INS1E. Figure 2 shows the staining of REST and insulin. We see in the REST mice 5, high level of REST is directly proportional to high levels of cell death. This is indicated by red color, which shows low levels of insulin production.
In one experiment, the pancreas of wild-type littermates and RIP REST mice were infused with glucose to measure levels of insulin production, as this would indicate levels of gene expression of target genes. You can see in figure 1a that wild-type mice, which are represented by the open diamonds, had normal levels of blood glucose with increasing time. However, the RIP REST mice, which are represented by the black dots, had much higher levels of blood glucose increasing over time. In figure B, the insulin response to glucose was measured. The black dots representing the RIP REST mice, and virtually no insulin was secreted in response to increasing glucose. However, wild-type littermates had a normal response. You can see in figure A that the RE1 containing genes were identified using a national database. The genes identified had anti-apoptotic properties and a conserved RE1 sequence. In figure B, they used quantitative polymerase chain reaction of RNA in INS1E cells. Expression of several genes, including ones coding for IRS2, PTPRN, CDK5R2, NRG, delta-2 catenin, and apoptosis inhibitor 5 were reduced after expression of REST. In figure C, what it represents is they verified whether these genes were downregulated by REST. Islets of RIP REST mice were analysed, and expression levels of several REST genes were reduced as a result of this. In figure D, CDK5R2 was decreased upon REST expression. This is represented by immunoblotting, and lower levels of CDK5R2 are detected with high levels of rest. Infusion of the pancreas with glucose in both wild-type littermates and RIP rest mice led to results that showed the wild-type mice had a normal insulin response to glucose, as opposed to the RIP rest mice, which had no response and led to severe hypoglycemia and fatalities. Quantitative real-time PCR analysis of islet mRNA indicated that the expression of the rest transgenes downregulated the expression of several genes essential for insulin exocytosis, including CDK5R2. Rest prevents the transcription of a number of genes that are essential for insulin production by suppressing anti-apoptotic genes. Numerous genes are affected by rest, but most significant is CDK5R2, which is a gene encoding a cyclin-dependent kinase activating protein, which is involved in insulin production, and IRS2, insulin receptor substrate 2, which encodes for the IRS2 protein involved in regulating receptor tyrosine kinases. These results have high biological significance for producing new medications for diabetes, which work by targeting the repressor element silencing transcription factor, thus allowing anti-apoptotic genes to be transcribed. Thank you for watching our presentation.